Well, first of all, I would like to thank Jose Carlos and Sabrina and the rest of the organizing team for inviting me here. I'm a tropical medicine researcher. I don't work on marine ecology, so this talk is going to be a bit different. So this talk today I title is From Field to Bench, and from bench to field. Marine biodiversity and tropical medicine research is working together. And I hope that after my presentation, you know I titled this like this. So if we look at the research interests of the Biocomat team, if you look at the webpage, which I've been copying yesterday, it says that the research is ultimately aimed towards the understanding of biological interactions and physical processes that drive the structure and dynamics of marine populations and communities in subtropical North Atlantic ecosystems. Wow. And there's us, the Tropical Diseases Institute. We're just interested and we focus on the diagnosis of pathogens in patients, development of novel therapies, and we study neglected and emerging diseases. So what's the link? Are the tropical diseases in the marine world? Well, if we focus our talk to medicine, we have this statement what we always say, which is from bench to bedside. This is translational medicine, which basically means if we have some lab results that we could apply directly into the patient, we'll do it. For example, if we have a patient who's suffering from a bacterial infection and this bacteria is resistant to any kind of antibiotics, we could test in our lab which antibiotic we could use against it and also check whether they're sensitive or not to whatever we have. Sometimes this works, so we'll be from bench to bedside. But sometimes when we give them the result, it doesn't work on the patient, so it goes back to the bench. And we have the same situation from the other side. We'll be from the bedside, which the doctor is calling us, then back to the bench, and then back to the bedside again, because it happens that another pathogen appears, for example. So if we apply from bench to bedside to our collaboration, we could talk about the field, from bench to field, yeah? So what happened when we started to collaborate? That there was, if you've seen from Jose Carlos' talks, there were some mass mortality events happening in the Canaries, and we're mainly focused on the ones in Tenerife. So we wanted to know whether there was a pathogen involved. So what happens? Sea urchins are becoming our patients now. So as you saw yesterday, there were some mortality events going on. They started in Madeira in November 2009, and then there were some events happening all over the year 2010. So when we started to do some research, we focused on the mortality events happening in Tenerife and La Palma in April 2010. And as you saw yesterday, the populations of sea urchins, mostly diadema, were going very low. I got very impressed by this picture, for example, that Jose Carlos sent me, the solid skeletons in the seafloor. What was going on? When we look at the sea urchins, they seem to have what we could identify as a sea urchin ball disease. You can see the patches, they're losing the spines, and these patches are getting bigger and bigger, and then after some days, or some weeks, we're seeing the skeletons and debris. So when we started to check on this disease, because we're experts on humans and tropical diseases, we saw that most of the reported cases worldwide are due to bacteria, mostly the genera Pseudomonas, Euromonas, and Vibrio. So I said to Jose Carlos and Sabrina, let's check for these bacteria. So we collect some swabs from the lesions, from this baldness of the sea urchins, and we also see some coelomic fluid. But not only from the moribunds and the dead one, we also got some healthy ones and some patients with symptoms, just to check whether the bacteria was present in all of them. So what happened was that Vibrio colonies, yellow and green ones, were isolated from all the individuals. Here you can see one sample from moribund specimen. and some healthy ones as well. So the whole community seems to be infected. To further identify this bacteria, we did some PCR with universal primer pairs for bacteria and some DNA sequencing. And all the strains that we got were positive for species Vibrio genolyticus. This species is pathogenic to many marine animals, but they're also pathogenic to humans. So I screamed, Jose Carlos and Sabrina was like, wow, the whole community could get infected. Yay, we have a health problem. So from this path, what conclusions we could get so far? That Vibrio genolyticus seems to be a causative agent of sea urchin bull disease here in the Canary Islands. I'm basing this on the later test that Sabrina and Jose Carlos were doing. If we inject the healthy sea urchins with the bacteria, the disease appears, so they seem to be causing it. As I just mentioned, this is important because they're causative agents. These species of vibrios are causative agents of infections in humans and in other animals, so that's very concerning. We are also wondering whether this could be due to global warming and climate change, because we also monitor the seawater from the Canary Islands. And we've recently seen events of explosive growth rates of Vibrio species in the seawater. The later one was in this summer in Tenerife and Gran Canaria. In fact, there were some infections by Vibrio species in some humans. So this is what we have so far from this from field to bench. So now going to the other part of the talk, from bench to field. What can marine biology researchers could do with a molecular biologist like us that work in tropical medicine? Well, the link was there. We have the visit from Dr. Eva Dikova. She's a specialist, one of there's two specialists in the world. She's a specialist on pathogenic amoeba of marine invertebrates and fish. And she just arrived during one of these mortality events. Well, she's almost trying to retire now, but she said that she's not retiring until she isolates paramoeba species from all over the world. She wants a paramoeba species from each country in the world. So I rang my two friends, and they were like, can you go diving and collect some sea urchin for us, please? So they were very kind, and they got 109 sea urchins, healthy ones, and with signs of the disease. Most of them were diadema. So we took them to the lab. We started to extract the coelomic fluid, and we also got the gonads and fixed them and stained them just to check whether there were amoebas there. So isolating an amoeba, especially paramoeba, neoparamoeba, from these individuals seemed very easy, but it was a lengthy and arduous process. And because we couldn't use any antibiotics, it was very, very hard. So what did we get from this? This is all the poor dead sea urchins that we have to kill. All the moribunds, these results were only related to diadema because we only isolate amoebas in diadema. All the moribund sea urchins were positive for amoebae. Most of the strains were lost due to what I just mentioned, the bacterial overgrowth, but we were able to establish 8 strains, which I'm presenting here. They're quite similar in morphology. The locomotion was very similar, and the pseudopodia were very similar as well. So we thought they were almost identical. When we look at this morphology is compatible to the Paramoeba genus. And this genus, as well as Paramoeba, as commonly have the presence of a symbiont organism, Perkinseller-like organisms, which evolve with them. An important fact to establish neoparamoeba as well is that on the membrane, they don't have any microscales, which is something that has been established previously for the genus Paramoeba. So Eva was very disappointed because she really wanted Paramoeba, not neoparamoeba. So she wasn't happy, and we decided to do some phylogenetic analysis to see where we were wrong. We sequenced the small subunits of the ribosomal DNA of the A strains, but they all were Neoparamoeba branchiphylla. I was very happy because I isolated amoebas from sea urchins, but she wasn't at all. So in which situation are we now? We've gone from the field to the bench, from the bench to the field, and now we're back to it. Why? Because we're on the situation. We have Neoparamoeba branchiphylla species in moribund sea urchins. The event that we observed with all the mortality was similar to the one that Robert Shiplin described with paramoeba invaders. But there's something that is concerning us. Others have isolated amoeba from healthy specimens. Why not us? What's going on here? Then it's why I just mentioned this much mortality could be due to the sea urchins, to the amoebae. We don't know. We know that amoebas are opportunistic parasites, so we need to establish whether the infection is primary or secondary. As well, this sea urchin ball disease that we're seeing here could be a synergy between the infection of the amoeba and the bacteria. We need to carry on doing the experiments. What we do know, and this is a surprise for you too, is that the amoeba are not harboring Vibrio. We did PCR of these strains and they were all negative for Vibrio. So this is a step where we are now. And finally, just to acknowledge, obviously, the Biochem group health, Dr. Eva Dinkova Health, and my whole team at the University Institute of Tropical Diseases. So we're still looking for a cure for the poor sea urchin.